Researchers at the Technical University of Munich (TUM) have developed an automated analysis method for the identification and quantification of microplastics particles in the environment, in our drinking water or in foods. Microplastics are everywhere in the environment. The tiny particles with diameters of less than 5 millimeters can absorb and transport contaminants and toxins. The researcher says that there is an urgent need for analytical techniques to learn about the size, concentration, and composition of these particles. The TUM research team has developed a new process. To be able to detect microplastic particles, the researchers had several hurdles to overcome. The first was the problem of low concentrations. River water, for example, contains massive amounts of suspended solids and fine sand, with plastic accounting for less than 1% of the particles. These particles must first be isolated before their concentrations and ultimately their chemical composition are determined. Previous methods have relied on the analysis of the residues that are released when the samples are heated. With that approach, however, it is not possible to determine the number, size, and shape of the plastic particles. The new approach is particle-based. That means that instead of destroying the particles, the researchers analyze them directly. To do this, the researchers use a method known as Raman microspectroscopy. It works by shining a monochromatic laser source onto a sample and detecting the light scattered by the molecules. Comparing the scattered light against the laser source provides information on the substance under investigation. To analyze plastic particles with a diameter greater than 1 micrometer, they must first be filtered out of the aqueous solution, detected under the microscope, and then illuminated with laser light. Because plastics such as polyethylene, polystyrene, and polyvinyl chloride scatter the photons in characteristic ways, they each generate signals as unique as a fingerprint.